It's my pleasure to introduce our next speaker, Dr. Lewis Roberts, who needs no introduction here. He's a professor of medicine at the Mayo Clinic. He's been a leader in our field for many years and spoke to us earlier today about his GWAS efforts, but he's also an external co-chair for the TCGA. And so next he'll be talking to us about how molecular subtyping of cholangiocarcinoma links IDH mutations to altered mitochondrial biogenesis.
Thanks so much, Chip, and I'd like to thank the organizers for the opportunity to present as well. And what I thought I'd do was summarize some of the results from the TCGA project for cholangiocarcinoma and talk just briefly about cholangiocarcinoma and actually also briefly discuss some of the nuances of that data set and also some of the findings that are related to the IDH mutations within that data set. And at the end, if I have a little bit of time also, I'll describe some additional work that we've done with collaborators in Korea recently, looking into what Dr. Deshpande was actually telling us about the differences in the cholangiolar and the bile duct subtypes of cholangiocarcinomas. So TCG, or the Cancer Genome Atlas, was started around 2006 by the National Cancer Institute and the National Human Genome Research Institute with the idea of really accelerating our understanding of the molecular basis of cancer through the application of genome analysis technologies. And they started out with, I think, 3 cancers initially, but then expanded the project to try to profile up to 500 cases of each of the 23 major cancers. And then later on in the process, they added 100 cases each of 10 rare cancers. And again, I have to thank Stacy and the Cholangiocarcinoma Foundation because it was a joint lobbying effort to get cholangiocarcinoma added in as a rare cancer. But the idea then is that it produces comprehensive genetic, genomic, and proteomic analysis of the major cancer types and the rare cancers, and has really fueled a subsequent project, which is really the pan-cancer analysis of whole genomes, which is that of the about over 10,000 genomes that were generated, all of those that are being analyzed together as well, and there's insights that we are gaining from those efforts as well. And the goal, of course, is to stimulate applications of cancer genomics in medicine. Now that the main project is done, for example, there's sub-projects underway looking into immuno-oncology, looking into biomarkers related to all the different cancer types. And the data, I think, is one of the keys is it's broadly available to the cancer research community. And so up to, at this point, there's about 3,700 publications that reference TCGA data, and this is increasing very rapidly. So this just sort of summarizes the fact that we have, in all, 33 different tumor types with 10 rare cancers, about 11,000 patients contributed samples, and there were 7 different data types. And this, is an update on a slide that was from a couple of years ago. The bars in red were the TCGA projects that had the publications in press or published, and the ones in green were the ones that the analysis was underway. And what I did was put red stars where the publications have subsequently been published, several of them in 2017, including the projects for HCC and cholangiocarcinoma. So for the cholangiocarcinoma project, we actually ended up with cohort about 53 samples. We're able to do the complete analysis on 38 samples, and for 15 that did not pass the RNA qualification, we're able to do the whole exome analysis for mutations, and so we did the 15. And again, there's the because we weren't able to do the full analysis, TCGA wouldn't fund it, so the 15 were the sequencing of the 15 was actually funded by the Cholangiocarcinoma Foundation. So another kudos to the foundation there. So we're able to do the somatic copy number analysis on the 38, the whole genome methylation, RNA sequencing, microRNA sequencing, and reverse phase protein arrays in the set of 38. Now, in terms of the study cohort, some of the issues that TCGA had faced before was about bias from adjacent benign tissue that was not, in quotes, normal. So typically, in the case of cirrhosis, for example, if you have a patient with cirrhosis and liver cancer, and you get a piece of the liver cancer tissue and adjacent, in quotes, benign or normal tissue, that tissue already has molecular and genetic alterations. They were also concerned about the tumor tissue potentially having been modified by prior chemotherapy or radiation. So those things were typically exclusions. But what we found for the HCC project, for example, is that many source sites did not have germline blood DNA available, so tissue was the only available, in quotes, benign tissue available. And so in order to allow to have an adequate set of, to be assembled, they allowed up to 25% of samples for the HCC project that had no blood DNA control, to have benign adjacent tissue as long as it did not have full-blown cirrhosis. They allowed up to what we call F2 fibrosis. So we had detailed pathologic review of the TCGA cholangiocarcinomas, and this is just a summary in terms of their level of differentiation. So most of them were moderately differentiated tumors. And then, as I mentioned, we had the 38 total samples for all the platforms. Important thing to note here is that we actually submitted 83 samples, and we only had 53 that either met the full or the partial qualification. And this is because cholangiocarcinomas tend to have this high stromal content. And so because of, again, the rigors of the TCGA process, they insisted that the tumors have at least 60% tumor nuclei, because they wanted to be sure that there wasn't too much stromal contamination. But that meant that particularly for cholangiocarcinoma, as well as for pancreatic adenocarcinoma, we were limited in the types of samples that we could enroll. And I think that's important to recognize that, in a sense, there's a subclass of samples that this project did not analyze because of this inclusion criterion. But we ended up with a fairly homogeneous group of tumors. All 38 were non-liver fluke infected, so it allows us to do somewhat of a comparison with the data that we heard from Dr. Terry earlier today about the profiling from Thailand and Singapore. There were 37 of the 38 were free of hepatitis, so we only had one case of viral hepatitis, and 32 of the 38 had intrahepatic cholangiocarcinoma. There were 2 extrahepatic, 2 hilar, and we accepted 2 mixed ICC HCCs as well. And 37 of the 38 also were from Western countries. So this shows the basic analysis looking at the profile of DNA mutations. And this will be tough for me to see from this distance. I think I'm going to, this will be tough. I'm going to maybe just step up here and point out a few genes. Yep, a few genes here. So what we separated them into was, we separated them into NADH, oh, perfect. Thank you. So NADH metabolism, and this, of course, is what Dr. Kaden was just telling us about, and we heard about the IDH1 and 2 mutations. So we had a subset of, I think it was 7 of the 38 that were IDH mutant. And then we had BAP1 mutations, and the BAP1 is involved in deubiquitination, but also behaves fairly similarly to the chromatin remodeling gene. So several chromatin remodeling genes, ARID1 and 2, and related genes. We had the FGFR signaling aberrations, including several FGFR fusions, as well. And then other mutations, I'll point out here, changes in the CDKN2A or P16 gene, which is well known to be fairly heavily aberrant in this tumor. There were also MAP kinase signaling mutations, and so we talked about this earlier. There were some KRAS mutant tumors, but it's interesting because we had mostly intrahepatic cancers here and very few perihilar or distal. We don't see that many KRAS mutant tumors. We also had the PI3 kinase signaling pathway, pathways relating to histone methylation, the mTOR signaling, the Wnt pathway, Hippo signaling, and other miscellaneous genes as well. So we then set about to try to cluster these genes to see if we could order them into different subgroups or subtypes of the tumors. And one of the first things we saw, and this again has come up, is this preponderance of genes expressed in normal liver. What we found was that there was such a high expression of genes that are expressed in normal liver that it made it actually quite difficult to subclassify the cholangiocarcinomas. So the team actually excluded these genes, about 500 of them, and then did a subclassification based on all the other genes in the gene set. And so that then led us to this mRNA comparison, where you can see that we ended up with, and on the mRNA level, with 3 main clusters, clusters 1, 2, and 3. And interestingly, you'll see that the FGF receptor fusion tumors all were in the same cluster 3, whereas the IDH mutants were all in cluster 1. So just the independent clustering separated out these two subgroups of tumors. We also took this information of the 3 clusters then and went to a previous data set that Dr. Jesper Andersen and Snorri Thorgerson had developed at NCI. This was a collaborative effort that included some Mayo samples as well. And the 3 clusters, again, also subclassified with the IDH mutants all being in the cluster 1-like group. Not all of them, but mostly being in the cluster 1-like group. So we had sort of an independent validation of this finding. If we did a principal component analysis based on the RNA in these 3 clusters, you could see how in space the 3 clusters separated out as well. And then as we began to look then doing gene set enrichment analysis to see if we could find pathways of genes that were associated with the different clusters or different subtypes of these tumors, what we noticed, and much of this was done by Larry Kwong at MD Anderson, was that there was a difference in the enrichment for mitochondrial genes involved in oxidative phosphorylation compared to the genes involved in the chromatin modification pathways. So, sorry. So there were a number of genes that were shown then to be enriched in the IDH mutant set of tumors that all were related to metabolic and mitochondrial processes, whereas in there was downregulation in the IDH mutant enriched cohort in these chromatin and epigenetic modeling genes. And this shows that then as a heat map where we can see here here is, interestingly, we also found a number of hepatocellular carcinomas that had what we saw to be a cholangiocarcinoma-like phenotype, and we added them into this analysis. So here, the cholangiocarcinoma-like HCCs over here. You can see the FGF receptor 2 fusion tumors, the IDH mutant tumors, and then some of the chromatin remodeling genes. And in this case, looking at the mitochondrial gene signatures, you can see that signature is enriched in the IDH mutant and the PBRM1 mutant genes, and anticorrelates with the chromatin modifying genes. If we looked at a mitochondrial score, again, this mitochondrial score was high for the IDH mutant and the PBRM1 mutant, and low for the BAP1 and other chromatin modifying genes. And it's interesting, the FGF receptor 2 tended to group with the chromatin modifying gene sets. And we basically saw the same observation in Jesper Andersen's previous cohort as well. So this also seems like it might actually be a general phenomenon in cancer, and this goes back to discussions that we had, I think, last year about the whole oxidative phosphorylation and its potential and the role of changes in oxidative phosphorylation in cancer. And when Larry looked at all of the cancer types in TCGA, what he found was that looking broadly at oxidative phosphorylation high versus low, within almost every cancer type, he could find subsets of the cancers that were high or low in the OXPHOS signature. So going back to look at other ways that we could classify the tumors, we looked at copy number alterations, and we're able to subcluster into 4 different subclusters. So we divided them or described them as quiet atypical. So this quiet has very little chromosomal instability. This is looking at the chromosomes and ranking them by increased copy number or decreased or loss of or deletion. And you can see here there's very little change. Here you can see, for example, gain here of this probably is 1Q, and then loss of 1Q. And so here we had a group that we called genomically messy, a group here that seemed chromosomally unstable with frequent deletion of 6Q, 9, and 14, and then a group that frequently had amplification of the cyclin D1 gene region. So we ended up with 4 just based on chromosomal aberrations. Next, we look at genome-wide methylation, and here also we were able to identify 4 subclasses based on the genome-wide methylation analysis. And interestingly, as we've talked about before, one of the things that the 2HG does is that it substantially modifies gene methylation. So you can see that there's a cluster that really was almost completely IDH mutated tumors. And then there were clusters, there was a cluster that had most of the I think there was one that, well, these are chromatin modifying genes. They seem to be fairly scattered throughout here. Among the cholangiocarcinomas, like I mentioned before, most of them were intrahepatic, and that's the blues. We had a couple of perihylers, and then we had some distals, and the distals tended to cluster in this first methylation cluster here. So then we try to take all of this information and put it into one big clustering scheme and use the program that's called cluster of clusters analysis to do this. And what this allowed us to do then was to break it down into 4 groups. All of the extrahepatic cholangiocarcinomas grouped together pretty much into one of the clusters. Then we had an IDH-like cluster. We had this METH2 cluster that had most of the cyclin D1 amplified tumors. And then we had this METH3 cluster that had most of the FGF fusion or altered tumors, along with the BAP1 altered tumors together. There's one. There's one other thing I wanted to point out there, but I think I'll go on here. I think one of the other things that we observed then too was that the subgroups that we found were consistent with other studies that had identified subgroups in different sets of cholangiocarcinoma. So here's a couple of different sets of cholangiocarcinoma. This is the methylation analysis. And again, here we can see that the IDH group all sort of grouped together. And here the IDH group all grouped together. And this is Jasper's cohort again. Mostly, there's a couple of them that are outliers here. One of the interesting observations was that there seemed to be some interaction between IDH mutations and the ARID1A chromatin remodeling gene expression. And this is interesting here. So this is ARID1A gene expression here, and the level of methylation of the ARID1A promoter. And so what you see here is that those individuals who had mutations of the ARID1A, the stars here, of course, having had mutations of the ARID1A, they had expression of this mutant form mostly at relatively low levels. If they had, though, methylation or mutation of IDH, typically it caused increasing amounts of methylation of ARID1A and resulted in relatively low expression of ARID1A. And then we had over here the IDH wild types, which tended to have higher expression of ARID1A. So there seems to be some interaction between those two genes. This just sort of shows the region of methylation of ARID1A, and I'll go ahead. And then finally, we actually put together all of the 3 tumors that, in a sense, we tend to think of in the gradient from the liver through the bile duct to the pancreas. We put all of those 3 types of tumors together. And what we're able to show using the tumor map algorithm is that most of them actually separate out nicely with HCCs, separate from cholangios, separate from pancreatic ductal adenocarcinomas. But there was a few HCCs that actually mapped with the cholangiocarcinomas. And these were the same as the ones that we found where there's a small subset that had IDH mutations, and also some of them that some of them that had IDH mutations, and also some of them that had a cholangiocarcinoma phenotype without having an IDH mutation. So they had an IDH-like phenotype. So I think I'll skip this. This basically sort of summarizes that observation as well. So just to summarize from this integrated multi-omics is just the recognition that we ended up with these 4 subgroups, the extrahepatics grouping together, and typically showing KRAS mutations, IDH mutants grouping together with some of them clustering with the PBRM1 loss of function genes, the cyclin D1 amplification forming a group together, and then the BAP1 FGF receptor 2 group that also grouped together with in this cluster. So in summary, we found that there was sort of relative mutual exclusivity of the K-NRAS from the IDH1 and 2, reflecting again the gradient from intrahepatic through to perihilar and distal, and also for PIK3CA and TP53. IDH1 and 2 seem to partner with ARID1A, BAP1, and PBRM1 along with CDKN2A or P16 loss. The loss of these chromatin remodifiers is not mutually exclusive, suggesting that they may act together. And this, we hope, gives a preliminary basis for future molecular classifications. The IDH mutant and IDH mutant-like samples seem molecularly distinct. There was higher oxidative phosphorylation expression and mitochondrial DNA copy number. There was lower chromatin modifier expression, including ARID1A, ARID1B, and PBRM1, and a distinct copy number profile. And combined with the meta-analysis that we did, this suggested to us that the RAS and the IDH mutant groups form distinct subgroups. And that also epigenetic alterations are a hallmark of intrahepatic cholangiocarcinoma with DNA hypermethylation, mutation of key epigenetic genes, and regulation of epigenetic gene expression. And of course, the question about whether this tumor might be amenable to treatment with drugs that target the epigenome. So finally, I just wanted to have a couple of slides that relate to what Dr. Deshpande was telling us earlier about the different pathologic subtypes. So we have two data sets, one from the Dongshan Medical Center in Daegu in South Korea, and one from Mayo. And this is some unpublished data we have where we actually looked histologically and separated them into the cholangiola and the bile duct subtypes. So in Dongshan, you can see they had more of the bile duct subtype than the cholangiola. And when they classified them, they subclassified into two groups, A, that they described as group A and B, based on gene expression. And you can see, interestingly, that there may be differences in things such as CA19-9 and CEA expression as well between the two groups, and definitely in terms of KRAS mutation, more KRAS mutations in the B subclass, more IDH mutations in the A subclass. If we looked at the Mayo data set, this was fairly similar, except that we had fewer of the bile duct subtype here, more of the cholangiola subtype. But again, the bile duct subtype seems to be more likely to have high CA19-9s and KRAS mutations, with the cholangiola subtype more likely to have IDH or FGF aberrations. If we did principal component analysis in both cases, the Korea and the Mayo cohort, we could separate them for the two groups quite readily. And we interestingly then saw differences in outcome, that these were all patients who had surgical resection, but you can see here that the bile duct, the subtype had a better prognosis in general than the wait, I think this is the bile duct subtype, had a better prognosis in general than the cholangiola subtype had a better prognosis than the bile duct subtype. Yes. Now, if we go further to look at all the groups pooled together, we say basically the same thing. No? If we pool the two groups, looking at both the two groups together, again, the cholangiola subtype, which includes the IDH and the FGF receptor, did better than the bile duct subtype. And this is both looking at overall survival and the disease-free survival, time free of recurrence. So to summarize this, the histogenetic subclassification may predict survival outcomes of the resected patients. This, of course, needs to be validated with additional studies. And the second question that we've been trying to address is the question of whether this histogenetic subclassification might also predict response to therapy beyond the responses that we are seeing to IDH as well as to the FGF receptor inhibitors. Thanks very much.
Thank you very much, Louis, for a very comprehensive conversation.